Good morning, everybody. Welcome back to Daily Lectures from New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. I'm sorry I couldn't be back for a day or two because I wasn't keeping well. In fact, when my voice has changed, it was down with a little bit of fever. We're just trying to record what I can, right? So today we're back with the subject on molecular biotechnology on the third part of it, where it deals with eukaryotes. Now we're on the specific topic of engineering plant quality and proteins, right? It's got a couple of subtopics. I'll just show you the subtopics. Just hold on. Right, the subtopics are modification of plant nutritional content, amino acids, lipids, vitamins, iron, phosphorus, modification of food plant taste and appearance, preventing discoloration, sweetness, starch, genetic manipulation of flour, pigmentation, plants as bioreactors, antibodies, polymers, edible vaccines, plant yield, increasing iron content, altering lignin content, erect leaves, increasing oxygen content, summary. Sorry, I missed a topic there. Phytoremediation. Though we have the summary and review questions, right? So going back to the subject proper. We have modifications of plants' nutritional content. Right now, the modification of plants and nutritional content. Over the years, agronomists and plant breeders have been extremely successful in optimizing the useful properties, example protein or oil content, and increasing the productivity yield of a large number of crop plants. However, traditional breeding approaches to crop improvement are both difficult and slow, as they are intrinsically limited by the existing genetic content of crossbreeding strains. Conversely, the use of genetic engineering techniques allows scientists both to dramatically speed up the process of developing plants with improving characteristics and introduce traits that would otherwise be impossible to develop by traditional techniques. For example, on a laboratory scale, genetic engineering has been used to improve the nutritional quality of several different plants, including corn, maize, and pea, by modification of the amino acid content of some of the seed storage proteins to the fatty acid composition of both edible and non-edible oil-producing crops. And three, the taste of fruits and vegetables by the introduction of monolin, a sweet tasting protein, amino acids, seed storage proteins, which is used as sources of both carbon and nitrogen during seed germination, containing a limited number of amino acids which are organized into repeating peptide units. Often the nutritional value of these proteins is deficient because they lack one or more of the amino acids, usually lysine or methionine, that is essential for human health. The amino acid composition of the seed storage proteins can be altered to a limited extent by breeding programs, but genetic engineering strategies can also be used. And the bulk of the lupine, a grain legume that is produced annually in Australia greater than 800,000 tons, is used to feed cattle, pigs, and chicken. Unfortunately, like most other grain legumes, lupine is deficient in methionine and cysteine, and therefore lupine feed is supplemented with methionine to provide animals with more nutrition nutritious feed without methionine. Supplementation. Lupines are engineered to express sunflower seed albumin, which is both stable in the rumen and unusually. Where sunflower seed albumin escapes microbial breakdown in the rumen and is therefore available for digestion and absorption in the lower gastrointestinal tract. The transgenic lupine plants that express sunflower seed albumin were used as an animal feed and as expected rats that received transgenic lupines as their sole nitrogen source made significantly greater weight gains that did rats feed than did rats fed non-transgenic lupines, comparable to what would be expected if a non-transgenic lupine diet had been supplemented with pure methionine. One normal way to increase the lysine content of seeds is to increase the production of lysine in transgenic plants by deregulating the lysine biosynthetic pathway. The amino acids lysine, threonine, methionine, and isoleucine are all derived from aspartic acid. The first step in the conversion of the aspartic acid to lysine is phosphorylation of the aspartic acid. To lysine is phosphorylation of the aspartic acid by aspartokinase AK to produce beta-aspartyl phosphate, the condensation of aspartic beta semialdehyde with barbic acid to form 2,3-dihydrotypicolinic acid, which is catalyzed by dihydropicolinic acid synthase, DHTP, is the first reaction in the pathway inhibited by lysine. Thus, to overproduce lysine, it is necessary to abolish the feedback inhibition of these two enzymes. This was accomplished by cloning naturally lysine feedback in sensitive genes for DHDPS and AK from Corynebacterium and E. coli, respectively, fusing each of these genes to a chloroplast transit peptide to ensure that the two proteins are localized in seed plastids, placing each gene under the control of a seed-specific promoter and then introducing the two genes on a tri-plasmid binary vector into canola and soybean plants. Transgenic canola and soybean plants had more than 100-fold increase in the free lysine in their seeds, with an overall doubling of the total seed lysine content in canola and a 5-fold increase in the total lysine content in soybean. Currently, when corn is used as an animal feed, it must be supplemented with soybean meal, purified lysine, or both. In the future, it may be possible to replace the use of expensive lysine with inexpensive transgenic soybean meal than soybean plants that overproduce lysine. Moreover, it may eventually be possible by using the approach that has been successful with soybean to engineer corn to overproduce lysine. High lysine corn will be more nutritious for both animals and humans. Excuse me. On the lipids, it has been estimated that the annual global plant production will be worth around $70 billion by 2010. That's pretty much an older edition that I've been reading now. This is the 4th edition. More than 90% of this production is for human consumption in margarines, shortenings, salad oils, and frying oils. Together, soybean, palm, canola, rapeseed, and sunflower account for approximately 80% of the worldwide plant production. For the most part, these oils consist of palmitic, stearic, oleic, linoleic, and linoleic acids. In addition, some vegetable oils contain fatty acids with conjugated double bonds. This is in contrast to the more usual case in which the typical unsaturated fatty acid, polyunsaturated fatty acid of plants. Seed oils contain double bonds that are separated by methylene, CH2 groups. The presence of conjugated double bonds increases the rate of oxidation compared with polyunsaturated fatty acids with methylene interrupted double bonds, making them well suited for use as drying agents in paints and inks because they require less oxygen for the polymerization reactions that occur during the drying process. In an effort to eat healthier foods, consumers have been concerned about the nutritional content of various edible oils. As can be seen on Table 20.2, the fatty acid content of edible oils can vary dramatically. Moreover, it is deemed as desirable to have a low level of as possible of saturated fats, a high level of oleic acid, which lowers the undesirable low-density lipoproteins, or LDLs, without affecting the desirable high-density lipoproteins, or HDLs, and as high a level as possible of omega-3 fatty acids. Canola oil is considered to be one of the healthiest and most nutritious edible oils. In 1999, canola oil received an award from the American Health Foundation as the Health Product of the Year. Canola is derived from rapeseed, which may contain up to 40% erucic acid. However, following more than 40 years of conventional breeding, by definition, canola for Canada oil low erucic acid contains less than 1% erucic acid. Several reports have associated erucic acid with various cancers and heart ailments, and a maximum of 18 micromoles of all glucose, and a maximum of over 18 macromoles of all glucosinolates, secondary metabolites responsible for the bitter or sharp taste of many common foods such as mustard and horseradish, program and whole seed, notwithstanding the enormous success that has been achieved in developing canola by conventional breeding. It is currently possible by genetic engineering to rapidly modify a number of genetic traits to continue to improve canola. For example, approximately 90% of Canadian canola crop has been engineered to be herbicide resistant, and in addition, some varieties of canola have been engineered to be insect resistant. And it is currently feasible to change the degree of unsaturation, that is the number of carbon-carbon double bonds, and to modify the chain lengths of fatty acids in canola plants by genetic manipulation. The number of transgenic varieties of canola each producing a different modified oil have been created. Each transgenic variety contains one additional gene. For example, the production of shortening, margarine, and confectionery goods requires large amounts of stearate. A variety of transgenic canola contains an anti-sense copy of a brassica stearate desaturase gene. which inhibits the presence of the normal canola gene and leads to the accumulation of stearic acid rather than desaturation of stearic acid to oleic acid. Progress in the production of transgenic canola varieties with modified seed oil properties has been both rapid and impressive. The omega-3 and omega-6 fatty acids are precursors for many prostaglandins and are therefore directly responsible for regulating a number of important human metabolic functions. Until now, the major source for those important fatty acids have been marine and fish oils. However, for a variety of reasons, including the fact that global fish resources have declined dramatically in recent years, researchers have turned their attention to genetically engineered plants to produce safe, affordable, and renewable alternatives to traditional sources. Given the fact that plants can synthesize linoleic and alpha-linoleic acid, C18 precursors of the long-chain omega-3 and omega-6 fatty acids, it may be possible to engineer plants to transform linoleic and alpha-linoleic acids into long-chain omega-3, omega-6 fatty acids. In fact, when Arabidopsis thaliana plants were transformed in 3 separate stages with 3 additional genes, the linoleic and alpha-linoleic acids were converted to ractonic and eicosapentaenoic acid, respectively. The 3 introduced genes included a delta-9 specific longus from the marine microalga Isochrysalis galbana, a delta-8 desaturase from the protist Euglena gracilis, and a delta-5 desaturase from the fungus Martiarella albina. Despite the fact that the engineered plant contains higher than normal levels of arachidonic acid and eicosapentaenoic acid, that's about 7% and 3% respectively, in the leaf tissue, plant growth and development were normal. Thus, it is possible to engineer pathways in plants for the production of long-chain polyunsaturated fatty acids that are vital for human health. It is now necessary to engineer this pathway so that the 3 introduced genes are expressed in a seed-specific manner in an oilseed crop such as soybean or canola. Vitamin E, a substantial body of evidence, indicates that dietary supplementation with a lipid-soluble antioxidant vitamin E 400 international units, or approximately 250 mg of RRR alpha-tocopherol daily, results decreased risk for cardiovascular disease and cancer, assists in immune function, and prevents or slows a number of degenerative diseases in humans. The oils that are extracted from seeds have a relatively high level of total tocopherols, but in most cases only a very small fraction is alpha-tocopherol. Moreover, even in those instances where the fraction of alpha-tocopherol is large, example sunflower oil, to obtain a sufficient amount of vitamin E to confer the above-mentioned health benefits, it will be necessary for any individual to consume nearly 400 grams of oil daily. An alternative approach to this problem is to engineer plants to produce a greater percentage of alpha-tocopherol than gamma-tocopherol by transforming plants with a gene encoding the enzyme gamma-tocopherol methyltransferase, which catalyzes the addition of a methyl group to gamma-tocopherol. However, since a suitable gamma-tocopherol methyltransferase gene was not available, a strategy for its isolation had to be devised. One of the genes and the alpha-tocopherol biosynthetic pathway had previously been cloned from the plant A. thaliana. However, at the time, it was not possible to isolate the other genes in the pathway. This gene encodes the enzymes bihydroxyphenyl pyruvate deoxygenase HPPDase. A computer comparison of the sequence of HPPDase gene with the complete DNA sequence of the cyanobacterium Senecocystis sp strain BCC 6803 revealed the presence of an open reading frame that encoded a protein of the expected size that was 35% identical to the amino acid sequence of the Arabidopsis encoded protein. A Synchocystis putative HPPDase gene was found within a 10-gene operon thought to encode all the enzymes involved in the synthesis of alpha-tocopherol. One of the other genes within this operon encoded a protein whose predicted amino acid sequence was similar to those of several known plant delta 24-sterol C-methyltransferases. The enzyme has 5 adenosyl methionine binding domains, and as adenosyl methionine donates the methyl group to alpha to gamma tocopherol during the conversion. This enzyme has adenosyl methionine binding domains, and as adenosyl methionine donates the methyl group to gamma tocopherol during the conversion to alpha tocopherol. In addition, the gene had an N-terminal bacterial signal sequence where the cyanobacterial gene was cloned and expressed in E. coli. The recombinant protein catalyzed the methylation of alpha tocopherol to gamma tocopherol to alpha tocopherol. The DNA sequence of the Senecocystis gamma tocopherol methyltransferase gene was then compared with DNA sequences that were normally expressed in Arabidopsis and one Arabidopsis gene that showed 66% homology to the bacterial protein. At the amino acid level was identified, cloned, and expressed in E. coli. This recombinant protein converted gamma tocopherol to alpha tocopherol, the Arabidopsis gamma tocopherol methyltransferase gene, under the transcriptional control of a seed-specific promoter from carrots, was then used to transform Arabidopsis plants. The alpha tocopherol levels of these transgenic plants were significantly higher than they were in the non-transformed plants. Based on this model system, with the introduction of additional genetic manipulation, which increased the flux through the pathway, these results have been extended to corn embryos and soybean seeds, significantly increasing the nutritional value of the oils produced by these plants. Despite the progress that has been made, transgenic crops that produce higher levels of vitamin E are not yet sufficiently optimized for them to be commercialized. Vitamin A. Although rice sativa was a staple food of approximately half of the world's population, it is a poor source of several nutrients and vitamins, including vitamin A. About 124 million children worldwide are deficient in vitamin A. This deficiency leads to 1 million to 2 million deaths per year and is a leading cause of vision impairment, including night blindness and total blindness. One way to address the myriad of health problems that result from vitamin A deficiency will be to engineer rice to produce the vitamin A precursor provitamin A, beta-carotene. Mammals synthesize vitamin A from beta-carotene, which is a common carotene pigment normally found in plant photosynthetic membrane. In the year 2000, an international group of scientists reported using agrobacterium-mediated transformation to introduce the entire beta-carotene biosynthetic pathway into rice The phytoene synthase and the phytoene desaturase genes were introduced on a construct that did not contain any selective marker. The lycopene beta cyclase gene was a part of a separate construct that contained a selective marker. The frequency of insertion of all three genes into the rice genome and their subsequent expression were quite high. Thus, the engineered rice produces beta-carotene, which after ingestion is converted to vitamin A. At the time, it was thought that this strategy would facilitate the eventual development of transgenic strains of rice that not only produced high levels of beta-carotene, but also no longer contained any antibiotic resistance marker genes, removed as described in Chapter 18. Excuse me. The transgenic rice that produces beta-carotene has a yellow or golden color and has been called golden rice by the scientists who is involved in its development. Unfortunately, the initial version of golden rice, now called golden rice, one synthesized only 1.6 micrograms of beta-carotene per gram of rice, so that individuals would have to consume around 3 kilograms of golden rice, one each day, to reach the recommended animal daily requirement of vitamin A. The minimal daily requirement of vitamin A. However, in 2005, a scientist report has replacing the daffodil phytoene synthase gene with a similar gene from corn that produces an enzyme with a higher level of activity, resulting in a variety called golden rice 2 that produces 23-fold higher level of beta-carotene than golden rice 1. Moreover, in contrast to vitamin A, there are no harmful effects when individuals consume excessive amounts of dietary beta-carotene. The research that culminated in the development of golden rice was funded by several non-profit agencies and companies that hold the patents on the technologies that made the work possible have agreed to forgo the usual royalties. Therefore, the rice is expected to be freely available to farmers in the world's poorest countries. One of the problems for more widespread use of rice too in Asia has been a general mistrust of genetically modified foods by some consumers. In addition, both golden rice 1 and golden rice 2 were produced from subspecies Japonica cultivars of rice, which are produced with scientists but do not do well in the field in Asia. To remedy this situation, researchers are currently introducing the traits from golden rice 1 and golden rice 2 into the more popular subspecies indica varieties of rice by traditional genetic crossing of the indica strains with the engineered Japonica cultivars. As of mid-2008, the first field trial of an indica variety of golden rice was taking place at the International Rice Research Institute in the Philippines. It's hoped, following successful field trials, that golden rice can finally start fulfilling its promise. Folate. Tetrahydrofolate, or vitamin B9, is an essential micronutrient that is a necessary component of human diets. While the recommended daily intake of this vitamin is only 400 micrograms, the diet of many individuals in developing countries are chronically deficient in tetrahydrofolate. Tetrahydrofolate deficiency can result in severe birth defects, anemia, and neural tube defects. So, for approximately 3 billion people, most of them in developing countries, rice provides around 80% of the daily caloric intake. However, rice has a poor source of tetrahydrofolate as well as many other micronutrients. To increase the nutritive value of rice, the pathway for the production of tetrahydrofolate and tetrahydrofolate polyglutamate may be genetically manipulated. Folates are tripartite molecules, as they consist of 3 separate parts, including pteridine, p-aminobenzoic acid, and 1-homo glutamate molecules.
And folates are tripartite molecules, that is, they consist of three separate parts, including pteridine, P-aminobenzoic acid, one or more glutamate molecules. In plants, the pteridine precursors are synthesized from GDP in the cytosol. P-aminobenzoic acid is synthesized from KORAH-BIOTECH in the chloroplast, and then both the pteridine precursor and P-aminobenzoic acid are imported into the mitochondria. The final molecule is assembled and modified to express high levels of folate and rice, the FS genes in each of the chloroplastic and cytosolic pathways starting from KORAH-BIOTECH and GDP respectively were introduced from A. thaliana so that the two enzymes of these genes were overexpressed. One transgenic line at the level of tetrahydrofolate was approximately 100 times the level found in non-transgenic rice plants, which was more than sufficient to meet the recommended daily dietary amount of this vitamin. Another study by a different research group, similar genetic manipulations led to the enhancement of the level of vitamin in transgenic tomatoes. Iron. The World Health Organization has estimated that iron deficiency affects approximately 30% of the world's population. Is especially problematic where vegetable-based diets are the primary food source, although a number of crops are rich in iron. Absorption of this iron is often prevented by the phytic acid that's present in many other plants. As a first step toward developing food crops with sufficient rice plants to express the soybean protein ferritin. Ferritin is an iron storage protein that is found in animals, plants, and bacteria and carries up to 4,500 iron atoms in its central cavity. Excuse me. The soybean ferritin complementary DNA, cDNA, was cloned into a binary vector under the transcription control of the rice seed storage protein glutenin promoter. The entire construct was introduced into plants by electroporation. In this case, soybean ferritin was expressed in the seeds of rice plants and not in any other tissues. As a result, the iron content of the rice seeds per gram dry weight of tissue was increased to approximately 12.5 times the original value, while the iron content of leaves, stems, and roots did not change to any significant extent. Based on a typical daily adult portion of 150 grams of rice, the transgenic ferritin in rice provides approximately 30 to 50% of the recommended daily adult requirement of iron. While overproducing ferritin is an important first step in ensuring plants to provide additional dietary iron, it is necessary to ensure that the additional iron can be absorbed efficiently. To do this, rice plants were transferred into with three different genes. First, to increase the iron content, the rice plants were transformed with the ferritin encoding cDNA from green beans, Phaseolus vulgaris. Then, to improve the bioavailability of the introduced iron, plants were transformed separately with cDNA encoding phytase. Phytate is an inhibitor of iron absorption that can be removed by microbial phytase. Methionine, a family of cysteine-rich low molecular weight proteins that bind metals through the thiol groups of cysteine residues from the fungus of Aspergillus fumigatus and from rice respectively. To limit the expression of these genes to rice grains, the expression was controlled by an endosperm-specific promoter. Each of three different transgenic plants that were engineered produced the protein encoded by its transgene. Before these strains are crossed to generate a plant that can express all three of these transgenes and more, Heat-stable version of the phytates is required since cooking the rice resulted in the inactivation of around 90% of the phytates. Nevertheless, because obtaining sufficient dietary iron is so important to such a large number of people in the world, these preliminary results are exciting and promise to eventually have an enormous impact on the health of millions of people. Phosphorus. Most of the phosphorus in cereals and legumes is found in the form of phytate, phytic acid, or inositol hexaphosphate. Phytate cannot be digested by non-ruminant animals or by humans, so the absorbed phytate passes through the GIT, that's the gastrointestinal tract, and elevates the amount of phosphorus. In manure, the excretion of high levels of phosphorus can sometimes lead to environmental problems such as eutrophication, excessive plant and algal growth and decay. To increase the nutritional values of crops such as soybean and corn to non-ruminant animals such as poultry, swan, and fish, animal feed is generally supplemented either with phosphorus derived from rock phosphate or with phytates, which degrades phytate after it is digested. Unfortunately, such supplementation is expensive, adding significantly to the cost of producing these animals. However, beginning in the early 1990s, several low phytate mutants were isolated in corn, barley, rice, wheat, and soybean. In these mutants, an enzyme involved in one of the many steps is the conversion of glucose 6-phosphate to phytate. And this was typically altered so that the amount of phytate in the seed was often reduced by 50 to 90%. Moreover, the reduction in phytates was generally accompanied by an increase in inorganic phosphate, which maintained the total seed phosphorus level. The problem with this strategy is that the systemic reduction of phytate often has negative effects on the whole plant, resulting in decreases in seed germination, germ emergence, stress tolerance, and seed filling. Therefore, as an alternative to limiting the synthesis of phytate, one group of researchers constructed mutants of corn and soybean that were defective in the transport of phytate to seeds by silencing the ADP binding cassette ABC reporter in a seed-specific manner. The seeds of these plants had approximately in the level of inorganic phosphate. Moreover, the phytate levels in the rest of the plant were essentially unchanged, while these transgenic plants still need to be tested for their efficacy as animal feed. This approach promises to be important from both agricultural and environmental perspectives. On to modification of food plant taste and appearance. Preventing discoloration. The post-harvest discoloration of fruits and vegetables is a considerable problem for the food industry. A lack of acceptance of discolored foods by consumers has been dealt with by the food industry through the use of additives in a wide range of foods. However, the safety of some of these food additives, in particular sulfites, has been questioned. The enzymes that are thought to be responsible for the initial step in discoloration of fruits and vegetables, the oxidation of monophenols and delta ocyphenols, oquinones are polyphenol oxidases. These nucleus-encoded enzymes with a molecular weight of around 59,000 are localized in chloroplast with mitochondrial membranes. The contention that inhibition of the enzyme polyphenol oxidase would decrease the extent of discoloration has been tested with transgenic potatoes carrying a number of polyphenol oxidase cDNA constructs. Vectors are constructed with either the full length or partial potato polyphenol oxidase cDNA in either the sense or antisense orientation under the control of the cauliflower mosaic virus 35S promoter. The granule-bound starch synthase promoter or the patatin type 1 promoter. The last two promotes are specific for the potato tuber. The two commercial varieties of potato that were transformed with these constructs were considered to have a good level of intrinsic resistance to black spot enzymatic discoloration, so any increase of black spot resistance by genetic manipulation would be greater than that could be attained by traditional breeding techniques. Transgenic plants with polyphenol oxidase cDNA constructs were deliberately bruised, and then the extent of black spot damage was assessed. Most of the transgenic potato plants with an antisense version of the polyphenol oxidase gene under the control of either the cauliflower mosaic virus 35S promoter or the granule-bound starch synthase promoter was significantly more resistant to black spot than the non-transformed potatoes. The pethidine promoter, which may not be fully active in potato tubers, did not prevent polyphenol oxidase accumulation. The sense constructs all synthesized increased amounts of polyphenol oxidation and showed larger amounts of black spot than non-transformed control plants. It is hoped that these and similar antisense constructs will produce enzymatic discoloration in a wide range of commercially important plants. Sweetness. Even though a fruit or vegetable may have high nutritional value, if it is not tasty, humans usually will not eat it. All the palatability of food can be achieved by adding salt, sugar, flavors, or other ingredients. During preparation, it would be advantageous to the food industry if certain foods could be intrinsically more appetizing. Monelin, a protein that is found in the fruits of an African plant with the unlikely name of serendipity. This Coreophyllum cuminzidiales is approximately 300,000 times sweeter than sucrose on a weight basis. This feature makes manolin a candidate as a sugar substitute, with the added bonus that because it is a protein, it would not have the same metabolic impact as sugar. Manolin is a dimer with an A chain of 45 amino acids residues and a beta chain of 50 residues. The A chains are held together by weak non-covalent bonds. Unfortunately, the fact that manolin is composed of two separate polypeptide chains limits its usefulness as a sweetener because it is readily dissociated, denatured, and consequently loses its sweetness when it is either heated during cooking or exposed to acid, example lemon juice or vinegar. Also, the need to clone and express two separate gray genes in a coordinated manner complicates efforts to produce the protein in either transgenic or plants or microorganisms. To circumvent this problem, a manolin gene that encodes both the A and B chains is a single peptide which is chemically synthesized. The effusion gene was found in producing transgenic tomato and lettuce plants. Two different promoters were used to express manolin's fusion protein gene. In the experiment of tomatoes, expression was directed by the tomato fruit specific promoter E8, which is activated at the onset of fruit ripening. The construct for the lettuce experiment was under the control of the 35S promoter from cauliflower mosaic virus. Each construct used this transcription termination polyadenylation site from a type plasmid monoproline synthase gene. In each case, the synthetic manolin gene was introduced and plant cells by atymification infection used the type plasmid co-integrate vector system. Manolin was detected in ripe and partially ripe potatoes and in lettuce leaves, but not in green tomatoes. The manolin level in tomatoes could also be elevated by a burst of the plant hormone ethylene. This strategy for sweetening plants without sugar or chemical additives will be applicable to a wide range of fruits and vegetables. In addition to manolin, Several other potatoes have been reported. The genes for some of these proteins have been isolated, characterized. Given the demand for low-calorie sweeteners, as well as interest in healthy and natural food products, it is likely that many different plants will be genetically engineered to produce proteins to increase their sweetness. Fructans are naturally occurring polymers of fructose, but are not usually degraded in the human digestive tract. However, some beneficial bacteria in the human intestinal tract can utilize fructans. Small fructans, that is, up to 5 monosaccharide units, have a sweet taste and can be used as a natural low-calorie sweetener instead of sucrose. Fructans are generally produced from sucrose by fungal invertases on a large scale in an expensive process, or they are extracted from the roots of chicory plants or Jerusalem artichoke tubers. To develop an inexpensive means of producing fructans, researchers engineer sugar beet plants to convert the sucrose that they normally store in the vacuole of taproot parenchyma cells into fructans. This was done by transforming sugar beets with a genetic construct that contained the 1-sucrose fructosyl transferase cDNA from Jerusalem artichokes and the transcription control of the 35S promoter. To transform sugar beets, the genetic construct was introduced into protoplast in the presence of 220% polyethylene glycol. The transgenic sugar beets accumulate fructan up to 40% of the taproot dry weight. The mixture of fructans that was produced by the transgenic sugar beet plants is essentially the same as the fructans that were produced enzymatically. Biologically produced fructan may be an attractive alternative to the current industrial process and can be sold as either a non-digestible sweetener or as a health component that improves the intestinal flora. Starch. The starch that is found in most crop plants, such as potatoes, consists of 20% to 30% straight chain amylose and 70 to 80% branched chain amylopectin. The ratio of amylose to amylopectin has a large influence on the physical and chemical properties of the starch. For many, industrial application, it will be useful to have starch that is highly enriched in either amylose or amylopectin. Starch is normally synthesized in a stepwise process. The enzyme ADP, glucose pyrophosphate, phosphorylase catalyzes the transfer of ADP from ATP to glucose 1-phosphate to form ADP glucose. The enzyme which the enzyme starch synthase catalyzes the transfer of glucose from ADP glucose to the non-reducing end of a pre-existing glucan chain, which is a short version of an amylose chain by means of the alpha-1 full linkage. Branching can occur, catalyzed by starch branching enzyme, when two glucan chains are joined by an alpha-6 linkage to generate potatoes with a high percentage of amylose. The corresponding low percentage of amylopectin plants are transformed by using ace-attenuations with antisense versions of the starch branching enzyme under the transcriptional control of the 35S promoter and transgenic lines that exhibited only approximately 1% of the normal amount of the starch branching enzyme activity. The fraction of amylose increases from around 28% to 60 to 89% of the starch content. This is an initial step toward developing potatoes with unique starch. Compositions that may be used either for food or as a source of unusual starches with unique industrial properties. The freezing and thawing that are common in the development and use of frozen foods often lead to unwanted changes in the texture of the starch component, making the product less attractive to consumers. Excuse me. Under these conditions, the starch can separate into a solid phase and a liquid phase, a process known as syneresis. The long and branched amylose chains have a much greater tendency to form separate phases following freezing and thawing than the shorter branched amylopectin chains. One approach to creating a starch that could withstand freezing and thawing would be to modify the starch structure by decreasing the synthesis of amylose chains. This was achieved by transforming potatoes with antisense versions of 3 different starch synthase genes. This reduced the expression of each of these genes to very low levels and resulted in a dramatic decrease in the amount of amylose and at the same time in the length of amylopectin chains. These modified potatoes are currently being thoroughly tested to ensure that a variety of physical and chemical properties are unaltered so that they can be used in the production of frozen foods. Since starch is both inexpensive and very abundant, it is used in a number of industrial processes as a thickener, a gelling agent, or an adhesive. In addition, it is a substrate for the production of high fructose syrup and ethanol. The production of high fructose syrups requires the starch to be degraded, usually at high temperature, by the enzyme alpha amylase, and then treated also at high temperature with glucose isomerase, which converts the glucose released from the starch that was degraded by alpha amylase into fructose. The enzymes that catalyze these reactions account for a major portion of the cost of the industrial conversion of starch into fructose. Therefore, reducing the cost of the enzymes makes the overall process more attractive. With this in mind, researchers constructed a bifunctional enzyme containing the essential regions of both alpha amylase and glucose isomerase. Using the polymerase chain reaction (PCR) to fuse together the genes for these enzymes, of course, care was taken to ensure that the chimeric gene retained the correct reading frames of the 2 genes. When the above functional enzyme was synthesized, his E. coli, both alpha amylase and glucose isomerase activities were found, and both activities were identical to that found in the thermotolerant source organisms. The chimeric gene was introduced into potato plants under the transcriptional control of the granule-bound starch synthase promoter. Biochemical analysis performed on transgenic potatoes after treatment at 65 degrees centigrade, the optimal temperature for the alpha amylase, revealed a 3.9-fold and 14.7-fold increase compared with the non-transgenic potatoes in the concentrations of glucose and fructose, respectively, at 25 degrees centigrade. The glucose and the fructose concentrations were the same in the transgenic and non-transgenic potatoes. Thus, both enzymes function optimally at high temperatures and only very little or not at all at low temperatures. This work indicates that it is technically feasible to engineer potatoes to produce their own alpha amylase and glucose isomerase, and that the enzyme that is produced can then be used directly without the use or need for any purification in a process directed toward producing fructose. For many applications that employ plants containing high levels of starch, it will be desirable to be able to significantly increase the amount of starch produced by each plant. While it may be possible to manipulate the genes for some of the enzymes involved in the biosynthesis of starch, another approach to increasing the starch yield would be to increase the supply of ADP so that the flux through the first reaction of starch biosynthesis is increased. This may be done by decreasing the level of the enzyme adenylate kinase, which catalyzes the interconversion of ATP and AMP into ADP. To do this, a genetic construct encoding a plastic version of the adenylate kinase in an antisense orientation was used to transfer potato plants, resulting transgenic plants. The activity of adenylate kinase decreased significantly in leaves and tubers and dramatically in chloroplasts. Moreover, the transgenic plants show an altered pattern activity of the enzymes involved. the biosynthesis of starch. This lecture should be over in a minute from now, and we'll be continuing the rest of the portions in the next lecture. Please stay tuned. Best wishes. Thank you. A number of the transgenic plants showed an alternative pattern of activity of the enzymes involved in the biosynthesis of starch, and most importantly, a significant increase in both the yield of potato tubers and amount of starch in each potato tuber. This manipulation opens up the possibility of modifying other starch-containing plants in a similar manner. The next topic will be on genetic manipulation of flower pigmentation. But stay tuned, I will be back on the next lecture with the continuation of this topic, as this is a worldwide value of the flower industry. We'll deal in much more detail in the next lecture. Stay tuned, take screenshots, use them to the best of your studies. Thank you. And